Hello students. After determining DNA or protein sequences by sequencing methods, we compare these sequences to find out similarity or matches among these sequences. And that process of matching and to find out similarity is called as sequence alignment. The sequence alignment method is very important for structural analysis of the molecules as well as functional analysis of the molecules. So in this lecture, we are going to discuss what is sequence alignment, what are the different types of sequence alignment, and there are three methods of sequence alignment. Those we are going to discuss. So sequence comparison lies at the heart of bioinformatics analysis. It is a very important first step towards structural and functional analysis of newly determined sequences. So whenever you determine the gene sequence or a protein sequence by any of the method, the first step that we do in the bioinformatics is we compare the sequence with other sequences. And by this comparison, we come to know what is the relationship among these sequences. And that's a very important step for structural and functional analysis of this newly determined sequence. As new biological sequences are being generated at exponential rates, the sequence comparison is becoming increasingly important to draw functional and evolutionary inferences of new proteins with the proteins already existing in the database. So when you determine the sequence, if you want to understand about the properties of that sequence molecule, then you have to compare it with the sequences which are present in the database. Now, sequence alignment is a process by which sequences are compared by searching for common character patterns and establishing residue to residue correspondence among related sequences. So in this method, we compare two or more sequences, and the main objective of the sequence alignment is to find out a common character pattern present among those sequences. Now this common character pattern can be found out by establishing residue to residue correspondence. So you compare each residue of one sequence with the another sequence, and then you establish residue to residue correspondence, whether those residues are matching or not. In case of DNA sequence, the residues are nucleotides, and in case of protein sequence, the residues are amino acids. So you're try to figure out whether there is a match or mismatch among those residues. And in this way, you establish residue to residue correspondence. And once you finish through the entire length of the sequence, you get the common character pattern among those sequences. And this is called as sequence alignment. Then definition of sequence alignment. Sequence alignment is a way of arranging the sequences of DNA, RNA, or proteins to identify regions of similarity that may be a consequence of functional, structural, or evolutionary relationship between the sequences. So if two sequences, they are functionally related, means they are performing the same function, there are chances that those two sequences are similar in their constituent nucleotides or constituent amino acids. Then if two molecules, they share similar structures, then again there are chances that they are matching with each other. Or if two molecules, they are evolved from the common ancestor, means they are sharing some evolutionary relationship, then also the two sequences may be similar in their constituents, nucleotides, or amino acids. And in sequence alignment, we try to find out those regions of similarities. Now the sequence alignment is made between an unknown sequence and a known sequence, or between two unknown sequences. So the case may be you have one known sequence and one unknown sequence. You can perform sequence alignment among those two sequences, or you have two unknown sequences for which you don't have any knowledge. Still, for those two sequences, we can perform sequence alignment. Now, the known sequence is called as a reference sequence, and the unknown sequence is called as a query sequence. It involves the identification of correct location of deletion and insertion that have occurred in either of the two lineages since the divergence from the common ancestor. Now suppose there are two molecules which are sharing some evolutionary relationship. Now during evolution, mutation occur, and mutation may be of three types. It may be a point mutation, deletion mutation, or insertion mutation. So whenever you perform the sequence alignment among two sequences, you may identify the locations where some of the part of the sequence has been deleted or inserted. DNA and proteins are products of evolution. Now you all know that on the earth, life originates in the form of prokaryotic unicellular organism. Now that prokaryotic unicellular organism, it diverge evolutionary, it undergoes some mutation, and maybe two species have been formed from that single species. And thereby the process of speciation starts. The DNA undergo mutation, then subsequently the phenotypic characteristics got changed for some of the individuals of that population, and they might be diverged into the two species, right? So the molecules of DNA and protein that we have today, or the organisms have today, they are due to the process of evolution. The DNA sequences undergo mutation, and thereby new species might have formed. Now the building blocks in this DNA and protein sequences, they are nucleotide bases and amino acids respectively, and they form linear sequences that determine the primary structure of these molecules. Now during evolution, sequences gradually accumulate mutations and they diverge over time, and traces of evolution may still remain in certain portions of the sequences to allow identification of the common ancestry. So these DNA sequences, they may undergo the process of mutation spontaneously or non-spontaneously. So these mutations are continuously accumulated in the DNA sequences. And these accumulated mutations somehow result into the divergence of the sequences and divergence of the species. But the traces of this evolution, the traces of these similarities among the sequences may still remain in certain portions of the sequence to allow identification of the common ancestry. Now, the portion where these traces of evolution may still remain, they are called as conserved regions. The conserved regions are usually functionally critical. It means it is a site which is involved in the functioning of that molecule. Some of the residues that perform key functional and structural roles tend to be preserved by natural selection. Other residues that may be less crucial for structure and function tend to mutate more frequently. Now, this can be understood with the example of enzyme. The active site residues of an enzyme family tend to be conserved because they are responsible for a catalytic function. Now the active site amino acids, they interact with the substrate molecule and they convert it into the product. Now suppose there is a mutation at the active site residue, then that enzyme will no longer remain functional, right? And due to that, the active site residue, which are functionally critical, which perform the key function, it doesn't undergo the mutation. Or even if it undergo the mutation, then that molecule will not be selected by natural selection process. But the residues which are not taking part in the process of conversion of substrate into product, means the residues which are not the part of active site, they may mutate more frequently because they are not involving in the function of that molecule. Now, the degree of sequence conservation in the alignment reveals evolutionary relatedness of different sequences, whereas the variation between the sequences reflect the changes that have occurred during evolution in the form of substitutions, insertions, and deletions. So suppose two molecules, they are evolutionary closely related, means those two molecules have been diverged recently, they share an immediate common ancestor. Then in that case, the relatedness among those two molecules will be high, and you will find much more higher sequence similarity among those two molecules. And in case the two molecules, they are very distantly related to each other on the evolutionary time scale, means they are not sharing immediate common ancestor, but they have been diverged from each other maybe millions of years ago, then in that case, the sequence similarity will be very less among those two molecules. Maybe their length will be also different. So with the help of this sequence similarity or sequence alignment, we can find out the evolutionary relatedness among the molecules. It helps to characterize the function of unknown sequences. Now, suppose you are comparing an unknown sequence with a known sequence, and for the known sequence, you know about its structure and function and various other characteristics. If this known and unknown sequences matches with each other, they are showing significant sequence similarity, then you can predict the structure and function for that unknown sequence also, because it is matching with the known sequence. Then when a sequence alignment reveals significant similarity among a group of sequences, they can be considered as belonging to the same family. So suppose there is an enzyme molecules. Now those enzyme molecules, they are sharing significant sequence similarity, and their active sites, which are conserved sites, they are matching with each other. Then we can classify those enzymes into single family. So the sequence alignment can also help us to identify the sequences which belongs to the same family. Then sequence alignment can be used as a basis for prediction of structure and function of uncharacterized sequences. So again, if the unknown sequence is matching with some known sequences, and for known sequence you know its structure and function, then we can simply predict the structure and function of that unknown sequence because it is sharing some significant sequence similarity with the known sequences. If two sequences share significant similarity, the two sequences must have derived from the common evolutionary origin. Now, with the help of sequence alignment or sequence similarity, we can establish the evolutionary relationship among the sequences. If they are matching with each other, if they are sharing significant similarity, then we can say that they are sharing common ancestor. And if they are not sharing significant sequence similarity, then we can say that they are very diverse sequence. They are not sharing any kind of evolutionary relationship with each other. Then there are two terms in sequence alignment that we hear regularly. One is sequence homology and other one is sequence similarity. Now, when two sequences are descended from the common evolutionary origin, means they are sharing a common ancestor, they are said to have homologous relationship or they share the homology. So such a sequences will be called as homologous sequences. A related but different term in sequence similarity, which is the percentage of aligned residues that are similar in physicochemical properties such as size, charge, and hydrophobicity. Now this term sequence similarity, it indicates that what percentage of residues are matching or they are common among the two sequences. So suppose you are comparing two protein sequences of 100 amino acid long. Now out of those 100 amino acid long sequences, 80 residues are matching, is 80 residues are identical with each other, or 80 residues are similar with each other. Then we will say that the two sequences are 80% similar. Now that similarity we determine in terms of whether the two residues are sharing common physicochemical properties or not. Now this is applicable to protein sequences and not to nucleotide sequences, that is DNA and RNA sequences. These physicochemical properties of amino acids such as size, charge and hydrophobicity, that we have to look after whether they are matching with another residue or not. So sequence homology is an inference or it is a conclusion about the common ancestral relationship drawn from the sequence similarity comparison when the two sequences share high enough degree of similarity. So suppose there are two sequences which are sharing 80% similarity or 80% identity. Then we can say that these two sequences are homologous. So in a simple term, sequence homology is a conclusion that we derive from the sequence similarity analysis or sequence alignment analysis. You analyze two sequences, you align two sequences, you find out significant sequence similarity, then you can conclude that these two sequences are homologous. Remember, homologous means they are sharing common evolutionary ancestry or common evolutionary origin. The similarity is a direct result of observation from sequence alignment, and sequence similarity can be quantified using percentage, and homology is a qualitative statement. So as I mentioned earlier, that sequence similarity can be quantified in terms of percentage, like suppose 100 amino acid long proteins, they are sharing 80% sequence similarity or 80% residues are matching, then we can quantify it in terms of percentage. But sequence homology is not a quantifiable term. It's a qualitative term. They will be either homologous or non-homologous. For example, one may say that two sequences share 40% similarity, means 40% of the residues among two sequences are matching with each other. It is incorrect to say that two sequences share 40% homology. That is wrong. They are either homologous or non-homologous because it is a qualitative term, and that we determine on the basis of sequence similarity data. Generally, if sequence similarity level is high enough, a common evolutionary relationship can be inferred. So you compare two sequences, you find out higher level of sequence similarity among those two sequences, then you can say that the two sequences are homologous. If they are not sharing significant sequence similarity, let's say they are just 20% similar or 20% identical with each other, then we would say that they are non-homologous sequences. Nucleotide sequences consist of only 4 characters, and therefore unrelated sequences have at least a 25% chance of being identical. So as you know that DNA is just made up of 4 nucleotides A, T, G, C. So if you compare two unknown sequences, there are 25% chances that those two sequences will match with each other. But for protein sequences, they are made up of 20 possible amino acids. And so two unrelated sequences can match up 5% of the residues by random chance. Now, the shorter the sequence, the higher the chance that some alignment is attributable to random chances. So, you know that when you perform sequence alignment, they can be aligned in a two-way. Either there is a relationship among the sequences and that's why there is alignment, or the two sequences, they are unrelated to each other. They are not sharing any kind of homology, but they are aligning with each other by random chance. So the longer the sequence, the less likely the matching at the same level of similarity is attributable to the random chance. So shorter sequences, there are high chances that unrelated sequences will also match with each other. But if the sequences are long enough, then there are very less chances that the sequences will align with each other by random chances. Now in this curve, you can see that as the length of the sequence increases, the sequence identity chances are also decreases. So on the basis of that, there are 3 zones of protein sequence alignment. One is safe zone, other is twilight zone, and third one is midnight zone. So two protein sequences can be regarded as homologous if the percentage sequence identity falls in a safe zone, that is 30% or even higher. So if two sequences are 30% identical or more than 30% identical, we can confidently say that these two sequences are homologous. Then sequence identity value below the zone boundary but above 20%, they are considered to be in the twilight zone, where homologous relationships are less certain. So you have to perform some other type of analysis to determine homology among the two sequences if your sequence identity falls in the twilight zone. And the region below 20% is the midnight zone, where homologous relationship cannot be reliably determined. Then again, there are two terms which are different from each other. One is sequence similarity and sequence identity. So they are synonymous for nucleotide sequences. They are not different for nucleotide sequence. For nucleotide sequence, you can either say sequence similarity or sequence identity. But for protein sequences, these two concepts are very different from each other. In protein sequence alignment, sequence identity refers to the percentage of matches of same amino acid residue between two aligned sequences. So identical in case of protein means in one sequence there is a glycine and in other sequence also there is a glycine. Then we can say that these two residues are identical with each other. Similarity refers to the percentage of aligned residue that have similar physicochemical characteristics and can be more readily substituted for each other. So you know that amino acids are classified on the basis of their physicochemical properties into 5 classes. That is aliphatic amino acids, aromatic amino acids, then aliphatic polar uncharged, aliphatic polar positively charged, and aliphatic polar negatively charged. So these 5 different classes of amino acids are made on the basis of physicochemical properties of the amino acids. Those physicochemical properties are size, charge, hydrophobicity, etc. So if the amino acids belongs to same class, they will be called as similar because they are sharing common properties and they can be substituted for each other in the sequences. Now calculation of sequence similarity and identity. By this formula, we can find out the percent sequence similarity among the two sequences. The formula is S is equal to, in bracket, LS multiplied by 2 divided by LA plus LB multiplied by 100. So S is a percent sequence similarity that we want to calculate. LS is the number of aligned residues with similar characteristics, and LA and LB, they are the total length of each individual sequence. Then there is another formula to determine the sequence identity, that is I%. It can be calculated by formula I is equal to Li multiplied by 2 divided by La plus Lb multiplied by 100. Now here only one term is different, that is Li. Li is the number of aligned identical residues. In the upper formula for the similarity, Ls is a percentage of similar residues. While in case of calculation of I, that is percent identity, Li represent the number of aligned identical residues. Then types of sequence alignment. The overall goal of pairwise sequence alignment is to find the best pairing of two sequences, such that there is a maximum correspondence among the residues. To achieve this goal, one sequence need to be shifted related to other to find a position where maximum matches are found. Suppose there are two sequences which are different in their length. Now, why the length of these two sequences may be different? Because maybe in one sequence insertion mutation have took place, or in other sequence deletion mutation have took place. Now, in order to find out the alignment among these two sequences, you have to shift one of the sequence out of two in order to find out maximum correspondence or maximum alignment among the sequence. And this shifting will create gap in either sequence. And this will result into the maximum correspondence among the sequences, and this is called as optimum sequence alignment. So there are two different alignment strategies for two sequences. One is global alignment and local alignment. So the global alignment, Two sequences to be aligned are assumed to be generally of similar over their entire length. Now, this can only happen when two sequences are evolutionarily closely related, means they are sharing immediate common ancestor. Then and then only those two sequences may be similar over their entire length. And alignment is carried out from beginning to end of both sequences to find best possible alignment across the entire length between the two sequences. So in global alignment, we start the alignment from beginning to end. And in this alignment, we try to find out best possible alignment across entire length between the two sequences. This method is more applicable for aligning two closely related sequences of roughly similar length. For divergent sequences, the length of the two sequences may be dissimilar, and in that case, global alignment will not work because their length will be different as well as their sequence composition will be different. They must have accumulated more mutations as compared to related sequences. So in that case, we can use another method called as local alignment in order to find out the highly similar local regions between the two sequences. So this is the example of global alignment. We match two sequences from beginning to end and we establish common character pattern. Like in this case, you can see L to L match, K to K match, GG match, KK match, GG match. So we find out these common residues among the two sequences. And to enhance this alignment, to optimize this alignment, we have also placed gap somewhere. Like in one sequence, we have placed gap in between S and S. Then in second sequence, we have placed gap in between N and I. So if you do not place this gap, your sequence alignment or sequence similarity will be even less than this. So to maximize the chances of sequence alignment, to optimize the alignment, we should place gap among the sequences. So in the global alignment, we assume that the two sequences are of same length. They are similar over their entire length because they are sharing common ancestry. So the global alignment includes all the residues of the both sequences. In global alignment, the input is to treat the two sequences as potentially equivalent, and the goal is to identify conserved regions and differences among the two sequences. The application of global alignment is comparing two genes with same function. Maybe there is a gene performing same function in human and mouse. You compare that gene by global alignment method, or by comparing two proteins with the same function. So suppose there is a amylase enzyme you isolate from two different species. It is performing same function. Then you can compare it by the global alignment process or method. Then next is local alignment. Local alignment, on the other hand, it does not assume that the two sequences in question have similarity over their entire length. It only finds local regions with the highest level of similarity between the two sequences and align these regions without regard for the alignment of rest of the sequence regions. Now this approach can be used for aligning more divergent sequences with the goal of searching for conserved patterns in the DNA or protein sequences. The two sequences to be aligned can be of different length, and this approach is more appropriate for aligning divergent biological sequences containing only modules that are similar. So suppose there is a case that you have two sequences, but they are evolutionary highly distantly related. Means a lot of mutation have been accumulated in those two sequences. Now, when a large amount of mutation have been accumulated among those two sequences, they may be different in their length, they may be different in their composition, but if two sequences sharing homology, then the functionally critical site would have been conserved. And in local similarity, we try to find out that. So we do not compare sequence from beginning to end in the local sequence alignment. Instead of that, we try to find out the local stretches of similarity. And those local stretches of similarity indicate that this region among two sequences is conserved. Like in this case, you can see we do not compare the sequence from beginning to end. Instead of that, we try to find out the region of highest similarity among the two sequences. and that local region actually telling us that this is the region which is functionally critical, and that's why it is remain conserved. So the local alignment only include portions of two sequences that have highest regional similarity. So the local alignment, the input is two sequences may or may not be related, and the goal is to see whether a substring of one sequence align well with the substring of the other sequence. So we try to find out the local stretches of similarity. Note, for the local matching, overhangs at the end are not treated as a gap, and applications are searching for local similarities in large sequences, that is newly sequenced genomes, and looking for conserved domains or motif among the two proteins. Then again, on the basis of number of sequences aligning, there are two types of sequence alignment process. One is pairwise alignment and other one is multiple sequence alignment. So suppose we are aligning only two sequences with each other, it will be called as pairwise sequence alignment because it will form a pair. Two sequences will form a pair, and that will be called as pairwise sequence alignment. But suppose if you're aligning three or more sequences with each other at the same time, it will be called as multiple sequence alignment. So in pairwise sequence alignment, we only compare two sequences at a time. It is optimally is based on score. So when we align the two sequences, we score them. We give some kind of numbers for the alignment. And a pairwise alignment consists of series of paired bases, one base from each sequence. And there are three types of pairs. Either it will be a match or a mismatch or a gap. In the match, same nucleotide appear in both sequence. In the mismatch, different nucleotides are found in the two sequence, and in the gap, a base in one sequence and a null base in other sequence. So algorithms that we can use for pairwise sequence alignment are Needleman and Wunsch algorithm or Smith and Waterman algorithm, and programs that are usually used is BLAST, that is Basic Local Alignment Search Tool, and the result of BLAST is shown here in that picture. Then multiple sequence alignment. In multiple sequence alignment, three or more biological sequences, generally protein, DNA or RNA, they align with each other. So in pairwise sequence alignment, we align only two molecules at a time or two sequences at a time. But in multiple sequence alignment, we align three or more sequences at a time. And this is very important for phylogenetic analysis of the protein or DNA sequences. The programs that we use for this purpose, they are ClustalW, then PropCons or Muscle. And in this picture or diagram, you can see the result of multiple sequence alignment process. Then alignment algorithms. In mathematics and computer science, an algorithm is a step-by-step procedure for calculations. Algorithms are used for calculations, data processing, and automated reasoning. Alignment algorithm, both global and local, they are fundamentally similar and only differ in the optimization strategy used in aligning similar residues. Both type of algorithms can be based on one of the three methods, that is a dot matrix method or the dynamic programming method and the word method. So these are three alignment algorithm methods that we are going to discuss. One is dot matrix, dynamic programming and word method. Word method is also called as a heuristic method of sequence alignment. So first we will discuss dot matrix method. This is a most basic sequence alignment method called as dot plot method. Now it is a graphical way of comparing two sequences in a two-dimensional matrix. So in a dot matrix, two sequences to be compared are written in the horizontal and vertical axis of the matrix. The comparison is done by scanning each residue of one sequence for similarity with all residues in the other sequence. If residue match is found, a dot is placed within the graph. Otherwise, the matrix positions are left blank. And when two sequences have substantial region of similarity, many dots line up to form a continuous diagonal lines, which reveal the sequence alignment. If there are interruptions in the middle of the diagonal line, they indicate insertions or deletions in either of the sequence. And parallel diagonal lines within the matrix represent a repetitive region of the sequences. So in this example, you can see the two sequences to be aligned. We have written them on two axes of the matrix. So this is the first sequence that we have written in the horizontal axis, and this is the another sequence for the alignment that we have written in the vertical axis. Now the process of dot matrix method is that you compare first residue of one sequence with all residues of the another sequence. Like in this case, you try to match G with all these residues of another sequence. Here we find out a match, G to G match, right? Then there is no another G present in the sequence on the vertical axis. So we will not place gap here, dot here. Then let us see whether there is matching residue for A or not. So here we can see A match. So we will place the dot. A match, we will place the dot. A match, we will place the dot. So similarly, we do this process for all the residues present in the horizontal sequence, and thereby you will fill the whole matrix with the dots. Now you try to find out, or you try to establish a diagonal line among the dots which are adjacent to each other. Like in this case, you can see that these dots, if we join, we can form a continuous diagonal. Again, if we join this dots, we can find out a continuous diagonal. So wherever you find out a continuous diagonal, it indicates that these residues among the two sequences are aligning with each other, or they are matching with each other. Like you can see T, T, C, T, A, T. They are forming this diagonal. And you can find out the similar residues among the other sequence T, T, C, T, A. So these AT. So these are the residues which are matching with each other among two sequences. Again, here the diagonal is broken. So it indicates that in either of the sequence, insertion or deletion mutation have took place. Then there are these parallel diagonals you can see adjacent to this main diagonal. Now these parallel diagonals are indicating the internal repeat regions among the sequence. And those internal repeat regions are responsible for these parallel diagonals. Then a problem exists when comparing large sequences using the dot matrix method, namely the high noise level. So for DNA sequences, the problem is particularly very acute because there are only 4 possible characters in the DNA, and each residue therefore has a 1 in 4 chance of matching a residue in the other sequence. So to reduce the noise, instead of using a single residue to scan for the similarity, a filtering technique has to be applied which uses a window to fix a fixed length covering a stretch of residue pairs. So this is what the problem we get for large DNA sequences. Because the DNA sequence are only made up of 4 nucleotides, so there are chances that many of the nucleotides will match, and thereby a noise will create. Now here we cannot see a continuous diagonal where we can join the line, where we can join the dots and form the continuous diagonal line, because there are so many dots have been placed in this graph. So to reduce this noise, we can use a filtering technique. The filtering technique is called as a window. So in the previous example, we were matching single residue with all the residues in the another sequence. When we apply the window, when we apply the filter, instead of matching a single residue, we will match multiple residues at the same time. Let's say 5. The window size is 5. So instead of matching a single residue, now you will match 5 residue at a time with the another sequence. And if all those 5 residues matches, then and then only you will put a dot. Otherwise, you will not put the dot on the graph. That's the rule of using window or filtering technique. So by this way, we can reduce this noise. When applying filtering, window slides across the two sequences to compare all possible stretches. Dots are only placed when a stretch of residue equals to the window size from one sequence matches completely with the stretch of another sequence. The window is also called as a tuple. The size of tuple or a window can be manipulated. So you can set the size of the window so that a clear pattern of sequence match can be plotted. However, if the selected window size is too long, the sensitivity of the alignment will be lost. The dot matrix method gives a direct visual statement of the relationship between two sequences and helps easy identification of the regions of greatest similarity. Advantage of this method is in identification of sequence repeat regions based on the presence of parallel diagonals of the same size vertically or horizontally in the matrix. And it is useful in identifying chromosomal repeats. It can be used in identifying nucleic acid secondary structure through detecting self-complementarity of the sequence. Now, there are some web servers which will allow us to perform this dot matrix alignment of the two sequence. For example, there is a dot matcher and dot up. These two programs of emboss package, they have been made available online, and you can provide the sequence in the FASTA format, and the window of the specified length and the scoring scheme are also used in this dot matcher and dot up program. Then there is dot helix program. It is a dot matrix program for DNA or protein sequences. The program has number of options for length threshold and implements scoring matrices for the protein sequences also. Then there is a matrix plot. It is a more sophisticated program for alignment of protein and nucleic acid sequences, and the user has the option of adding information such as sequence profiles and distance matrices from known three-dimensional structure of proteins or nucleic acid. Then second method of sequence alignment algorithm is dynamic programming method. So in mathematics and computer science and economics, dynamic programming is a method for solving complex problems by breaking them down into simpler sub-problems. So if you have a complex problem, you simply break it down into simple sub-problem. Then you solve this simple sub-problem and you will get the solution for the complex mathematical problem. So dynamic programming is a method that determines optimal alignment by matching two sequences for all possible pairs of characters between the two sequences. So it performs rigorous mathematical calculations and it gives us the optimal solution or the accurate solution for the sequence alignment of two sequences. It is fundamentally similar to dot matrix method, but the dot matrix method was qualitative method where we simply put the dot. In case of dynamic programming method, it is a quantitative method where we actually provide the score for the match or a mismatch. Now in the dynamic programming, it works by first constructing a two-dimensional matrix whose axes are two sequences to be compared. So similar to the dot matrix method, here there will be two axes, horizontal and vertical, where we will write our two sequences to be compared. The residue matching is according to the particular scoring matrix. The scores are calculated one row at a time. And this starts with the first row of one sequence, which is used to scan through the entire length of the another sequence, and followed by scanning of second row. The matching scores are calculated, and the scanning of the second row takes into account the score already obtained in the first round. Now the best score is put into the bottom right corner of the intermediate matrix. So here there is a rule of dynamic programming. Now whenever you place the score for the A, it may be either match or mismatch, you have to consider the score which is coming from the adjacent cells. That is the cell from the upper side, the cell from the left, and the cell from the diagonal. That is X, Y, and Z. What you have to do, you have to add the maximum score from this X, Y, or Z direction into the score of A, and then you write the score of the A. So the score of lower right square A of 2 by 2 matrix is a maximum score from the one of the other three neighboring square plus and minus the exact single residue match score for A for the lower right corner and the gap penalty, that is GP respectively. So let us understand this by this example. We have two sequences ATTGC and AGGC. We want to align it with the dynamic programming method. So you write two sequences into axis. The first step that we have to perform is we try to match this first residue in the vertical section or vertical axis with all the residues of the horizontal axis. So AA match, we will write 1 score, AT mismatch 0, AT mismatch 0, 0, 0. Now when you start with the second row, you have to remember the rule of taking the maximum score from these three directions. So for G and A, there will be mismatch and there are no cell in the left as well as there is no cell in the diagonal, but there is a cell on the upper side of the vertical axis. So we will not consider the maximum score from that direction. But when you write the score in this particular cell, at that time you have to apply the rule of taking the maximum score and adding it into the uh, score of that cell. So the score of this particular cell is actually 0. So G and T, they are a mismatch. So the score of this cell will be 0. But the maximum score coming from three direction. This is 0, this is 1, and this is 0. So the maximum score is coming from the diagonal axis, that is 1. So 1 plus 0, we will write here 1. And similarly, we have to fill all the positions for this matrix. So simple scoring system is applied in which an identical match is assigned a score 1 and a mismatch is assigned a score 0. The score in the matrix are filled one row at a time and one cell at a time beginning from top to bottom. The best scores are filled to the lower right corner of the submatrix according to this rule. So you fill the entire matrix in this way, and when all the cells are filled with the score, the best alignment is determined through trace back procedure. So this is the final procedure that we perform here, trace back procedure, to search for the path with the best total score. When the path moves horizontally or vertically, the penalty is applied. So you can see you start with the last cell and you go to the first cell of the matrix, and the path you choose is such that the summation of the scores in each cell will be maximum. So you get this kind of final alignment AA, GG, and CC. So this is what the final alignment that we got from the dynamic programming method. If two or more paths reach the same highest score, one is chosen arbitrarily to repeat the best alignment. The path can also move horizontally or vertically at a certain point, which corresponds to the introduction of gap or insertion or deletion for one of the two sequences. So whenever you move horizontally or vertically, you have to provide a gap in either of the sequence, and thereby we can get the optimum alignment. Now this dynamic programming method is called as exhaustive type of algorithm because it uses a rigorous mathematical calculation to find the best or exact solution for the particular problem by examining all mathematical combinations. And dynamic programming is an example of this exhaustive method, and it is computationally very extensive. It will take a lot of time for the computer to perform this type of alignment. The third type of alignment algorithm is called as a word method or heuristic method. Now this heuristic method, it is a computational strategy to find an empirical or near optimal solution by using certain rules of thumbs. So here in heuristic method, instead of performing those rigorous mathematical calculation, you use some rule of thumb and you take a shortcut so that you get the optimal solution in a reasonable amount of time. Essentially, this type of algorithms takes shortcut by reducing the search space according to some criteria. However, the shortcut strategy is not guaranteed to find the best or most accurate solution. It is often used because the need for obtaining results within a realistic time frame. without significantly sacrificing the accuracy of computational output. So suppose in dynamic programming, which is an exhaustive type of algorithm for the particular sequence alignment or for particular database similarity searching, the computer is taking maybe 3 to 4 hours. If you use heuristic method instead of dynamic programming, then the same task will take around few minutes. So within few minutes, the results will be on your screen. So heuristic method, it is taking shortcut. It is not providing you the accurate solution, but it is providing near to accurate solution in the realistic time frame. So for database similarity searching, usually we depend on the heuristic method. And when we want to refine the alignment, when we want more accurate alignment, then we depend on the dynamic programming type of exhaustive type of algorithms. So these are three different methods for sequence alignment algorithm. One was dot matrix method, other one was dynamic programming, and the third one is a word method or a heuristic method. And on the basis of word method or heuristic method, two programs have been constructed. Very popular computational tools are available, that is FASTA and BLAST. So that's all for now. Thank you very much.